Hi, today I will be giving you a tour of the animal cell. Please feel free to ask questions if you have any. I am your tour guide and my name is Jericho Curry. We are starting off today by heading on into the cell. As we are inside of the animal cell now, coming up on the right hand side are ribosomes. Ribosomes are particles made of ribosome, RNA, and protein. The function of this organelle is to carry out protein synthesis in two locations. The two locations are the cryostat, which is free ribosomes. Another location is on the outside of the endoplasmic reticulum of the nuclear envelope, which is known as bound ribosomes. If you look, this is the bound ribosomes, which is located on the outside of the endoplasmic reticulum of the nuclear envelope. Now, let's continue. As you look straight ahead, come over to your left hand side, there's the Golgi apparatus. The Golgi apparatus is a membrane structure with a single membrane. Golgi modifies products of the endoplasmic reticulum, manufactures certain micromolecules, and also sort and packages materials into transport vesicles. Golgi is known as the stack of membrane-bound vesicles. Now we are about to make a U-turn. In about a mile ahead, you'll see lysosomes. Lysosomes are common in the animal cell. Lysosomes are a membranous sac of hydrolytic enzymes that can digest micromolecules stomach of the cell. Lysosome enzymes can break down proteins, fat, polysaccharides, and nucleic acids. Going forward, we are about to pass the mitochondria over there by the free ribosomes. Mitochondria are organelles that release energy from stored food molecules. Mitochondria are the sites of cellular respiration of metabolic process that generates ATP. Mitochondria use energy from food to make high-energy compounds that the cell can use to power growth. Mitochondria basically provide the energy a cell needs to move, divide, produce secretory products, contract, in short, they are the power center of the cell also. Now we are about to head over to the nucleus. But before we get to the nucleus, we must go through the nuclear envelope and then through the nucleus. Now we are currently approaching the nuclear envelope. The nuclear envelope is a double membrane layer. The envelope is dotted with thousands of nuclear pores, which that allows material to move into and out of the nucleus. The next section that we will be going through today as we continue is the nucleus. The nucleus controls most cell processes and contains the hereditary information of DNA. The nucleus is enclosed in a double membrane and communicates with the surrounding cytosol via numerous nucleus pores. Inside of the nucleus you will find the nucleus. Alright guys, we're heading inside of the nucleus. To get to the nucleus, the nucleus is a permanent structure in the nucleus. The nucleus produces ribosomes, which move out of the nucleus and takes position on the rough endoplasmic reticulum, where they are critical in protein synthesis. Now we're about to head onto this highway, I-65, on over to the smooth endoplasmic reticulum. The smooth ER is throughout the eriotic cell, and it gets its name because apparently it appears smooth by electron microscope. The smooth ER functions are depending on the specific cell type, including lipid and steroid hormone synthesis, breakdown of lipid-soluble toxins in the liver cell. It controls the calcium release in muscle cell contraction. As we are getting close to the end of this tour, we will have to make our way over to the vacuole. The vacuole is a membrane-bound sac that plays roles in the intercellular digestion and the release of cellular waste products. Vacuoles are generally small in an animal cell, but bigger in a plant cell. There are only a couple more stops to go, you guys, so let's keep exploring the animal cell. Smile! We are off to see the wizard, just playing you guys, but we're off to see the centrioles. Each centriole is a ring of 9 groups of fused microtubules. In each group there are 3 microtubules. Centrioles are a part of the cytoskeleton. The centrioles in an animal cell are arranged perpendicular to each other. Next stop of this tour is the microtubules in the microfilaments. Microtubules and microfilaments are two of the three types of molecule structures. It is also a part of the cytoskeleton fiber network. Microtubules are hollow tubes. Its wall consists of 13 columns of tubular molecules. Its main functions are maintenance of cell shape, cell motility, chromosome movements, and cell division and organelle movements. Microtubules are the thickest of the three components of the cytoskeleton. Now, microfilaments are a part of the cytoskeleton fiber network as well. It's one of the three molecule structures. The microfilaments structures are the maintenance of shell shape, changes in cell shape, muscle contraction, cytoplasmic streaming, cell motility, and cell division. We are now off to a chromatin. The chromatin is located inside of the nuclear envelope. Chromatin consists of DNA and proteins and condenses to form chromosomes. Next up, as we are exiting the animal cell, coming up on your right hand side, you're going to see the cell membrane, which is also can be known as the plasma membrane. The cell membrane is the outer part of the animal cell. It's the first thing you run into as you enter the cell, and it's the last thing you run as you exit the cell. The cell membrane is a vital component of cellular control. Its responsibility is to restrict what can enter or leave the cell. The process of this transaction is called homeostasis. I hope that everybody enjoyed this animal cell tour. I hope you came with nothing and you are leaving with a lot of knowledge than what you came with. Also remember what the cytoplasm is. The cytoplasm is the substance that fills the cell. It's a jelly-like substance. Also, the cytoplasm is a part of the cytoskeleton. As you look all around the cell, what each organelle is surrounded by is cytoplasm, which is the jelly-like substance. Also remember, my name is Jericho Curry, and I was your tour guide for today for the animal cell.